right then, we can get started. I have a couple more slides for the protein lecture and we'll move on to carbohydrates. So just a reminder for the recap questions where we at. We talked about Dumas and Kildare. Last time we went into details and differences between the methods. Okay, who can summarize for us the principle of Dumas method? Just in few words. What is it based on? Anybody would like to volunteer? Those of you online? We have few here, but I feel like I always look at them and they feel that I'm cornering them. Anybody can answer. Yes, very simple. It's a method based on nitrogen combustion. So use a nitrogen analyzer and the food material is combusted at really high temperature and we just scrub everything off, all the gases, and what we send to the detector is only the nitrogen, and it is detected by thermal conductivity detector. You will learn more about it in lab next week and analyze your different food products using the Dumas. Okay, we talked about Kildare in a lot of detail. So can you compare both? Compare and contrast. You don't have to list all the differences, but maybe one similarity, one difference that you can think of between the two. Let's see. Okay, I'm starting to see some answers here. Yes, and Kaldal is instrument-based. Well, sorry, is titration-based, volumetric. That is very true. And LICO is based on weighing your sample and getting, it's not really gravimetric. You do not get a weight of nitrogen by actually physically measuring. No, it is combustion. So we cannot say it's gravimetric. We can say that Kaldar is volumetric. That is true, but it's not necessarily, the Dumas is not, you take a weight and then you take a weight at the end. There is no such thing in Dumas, so we cannot call it a gravimetric assay. We actually determine the conductivity of nitrogen. So it's not about the weight, and we convert that to a weight by having a standard curve. Okay, so others, I saw Dumas is instrument-based, Kaldal is mostly reagent-based, Kaldal is cheaper, yes, and it's low maintenance compared to the Dumas method. Dumas method is free of reagents, so technically safer. In Kaldal, you use concentrated H2SO4, concentrated NaOH, so you use corrosive reagents. Both are based on nitrogen analysis. So we're analyzing nitrogen content and converting that to protein. Both are very precise and most of the time accurate. And they're both official methods of analysis. Okay. So you can review the slide of comparison. There is one in here somewhere. So far we talked about the Dumas, the Kildare. We mentioned infrared. We talked about BCA and Bradford. The Burette method and Lowry method are also based on reducing cupric to cuprous. Using the reduction power of proteins and specific amino acids in the protein. So they are just kind of a diversion of BCA. BCA is more, is a newer method than Burette and Lowry, and it kind of dealt with some of the inconsistencies of these other methods. But all of these methods listed are research methods. Like I said last time, they're not really used to accurately quantitate protein in a complex matrix, mostly in a solution where we purified the protein, and we need to determine how much we have in this protein in solution for a different assay or for specific activity. Another research method that I'll talk about briefly is the UV detection method. So absorption at 280 is mostly defined by the aromatic amino acids. So there are three: tyrosine, tryptophan, and phenylalanine, but phenylalanine does not have high absorptivity as tryptophan and tyrosine. So most of the absorbance at 280 comes from tyrosine and tryptophan absorption. So proteins in general, they have a different concentration of amino acid, but all proteins have to some extent tyrosine and tryptophan in them. But if you're measuring hydrolysates, like if you have a protein that has been digested and you have some peptides that don't have tyrosine and tryptophan in them, so the protein has been chopped off and some peptides made up of, let's say, 5 to 10 amino acids or more or less, they might not have tyrosine and tryptophan, so they cannot be detected at 280. In this situation, we can measure absorption at 220 or around 220 plus or minus few nanometers there. So that 220, we have absorption of the peptide bond at this range. So if we think we have peptides, we can measure at 220. So we can determine concentration if we know the molar extinction. Remember in Beer's law, absorption equals absorptivity times the distance in the cell, the measuring cell, times concentration. So if we know the absorptivity of the molecule, we are determining, or the compound we're determining, then we have the absorption, we have the absorptivity, we have the length as constant, we can determine concentration. But often we go with standard curves. So commonly used is bovine serum albumin, is a common protein used for determining concentration of proteins using assays such as UV absorption. So you prepare standard, prepare them at different concentrations, and you measure absorption, you plot the standard curve, and you get the concentration using the equation of the line. So it is non-destructive. So if you have a little bit of sample and you need to use it for some other assays, you can, because you just measure the absorption and take out the sample. It's not destructive. Whereas Keldal and Dumas is destruction. You cannot reuse your sample for anything else. So that is a good thing. And also for BCA, you're adding reagents, there are interactions, whether it's Bradford, the same thing. So these all are destructive methods. This is non-destructive. It's really nice to have an option where you can use the sample for multiple assays. It's very rapid. That's another advantage. You don't have to add any reagents. There's nothing. If you have a clear solution with protein, it's easy. You measure that right away. You measure absorption right away. So again, it has to be pure and clear solution. Clear solution is more important. Even if you have other components in there, like sugars, you might have reducing sugars in there. And that's where BCA won't work because BCA is based on reduction. So you'll get interference. But here, if it's a pure, if it's not necessarily 100% pure, but it's clear and no interference, that means you don't have any other components that can absorb at 280, then this would be a good method to follow. So that's the end of the protein here. Any questions about anything? Okay. We'll move on. Carbohydrates. So here in this section, it's it's not part of proximate analysis. So proximate analysis is over. So we have to think about proximate analysis is the quantification of ash, protein, fat, and moisture. And by difference, you get the total carbohydrate. We're done with that. Here, we're going to talk about actually analyzing different carbohydrates in our sample. Okay, so it's carbohydrate analysis is not part of proximate analysis. Okay, good. Which of the following is a carbohydrate or are carbohydrates? Yeah. What's that? D. Sam goes with D. Any other question? Any other answers? Oh wow, people are answering D. Do you all agree? You must have seen the next slide. Oh, I'm getting different answers. G. Yeah, I like to see G because it's the wrong answer. So A and B are carbohydrates. Lignin is not. All of them are dietary fiber components. So dietary fiber does not mean that it is a carbohydrate. So lignin is not a carbohydrate, it is a dietary fiber. So this is what a lignin looks like in terms of structure. The components, the subunits, are not carbohydrate subunits. So they're not glucose or anything that has a carbohydrate subunit. So they're alcohol subunits. So that's why they're not carbohydrate, but they're not digestible by our body. So that's why they are in the category of dietary fiber and specifically insoluble dietary fiber. Any guesses there? The method used to determine starch gelatinization could be used to determine starch retrogradation. Okay, in order to be able to answer this question, you need to know two things. You need to know the difference between starch gelatinization and starch retrogradation, which is a food chemistry topic, and you need to get this lecture. So I don't expect you to know if it is the same method, but I expect you to know the difference between starch gelatinization and starch retrogradation. So who is brave to tell me the difference? Al. Yes. So Al described it in a very simple and correct manner. So starch gelatinization happens when we heat our sample product, ingredient, whatever it is that has starch in it, that is uncooked starch. The starch granules will swell because moisture is going to go in. And that's what, when moisture gets in and they absorb moisture, that's the swelling process that leads you to gelatinization. So that requires heat and some time to reach gelatinization. And to get starch to be digested in our gut, it has to be gelatinized. Raw starch resists digestion. Retrogradation, it happens after we cool down. So after the processing is over and we cool down, and then during storage, the starch, amylopectin and amylose start to recrystallize with each other, and they form retrograded starch. So retrograded starch also resists digestion. So speaking of resist digestion, do you remember from food chemistry the four different components of resistance starch? resistant starch become part of dietary fiber. And the higher that resistant starch you have, that they say is good for you because it lowers the glycemic index, especially in people that are diabetic, because you don't release glucose fast. So, do you know what the difference? I already said retrograded starch is a resistant starch. I already said raw starch is also in the resistant category because we cannot digest raw starch or efficiently digest raw starch. So these two, can you remember two others? The people are quiet online. Anastasia? Yes, chemically modified starch, it can resist digestion, so that's a good third category. Can you remember a fourth one? That's the one that people mostly forget. It's the starch that is embedded physically entrapped and the enzymes can't access it. Okay. So now back to this. Is it true? Is it false? I'm going to tell you it's true. So you can mark it true. And later on, not today, we won't get to it today. The same method to measure starch gelatinization, we use it to measure starch retrogradation. Why it is important? It is important for product development and understanding the impact of processing, the impact of storage on changes in starch. So it's important to measure starch gelatinization and starch retrogradation. And an example of starch gelatinization and retrogradation is bread. So when you bake the bread, the starch is gelatinized, it helps in the formation of the structure of the bread, and also helps with us being able to digest the starch. But over time, the bread becomes stale. And part of that staling process is the degradation of the starch, contributing to that staling process. So how the texture changes of that bread. Okay. Why is it okay? Before why it's important to analyze carbohydrate, what's the importance of carbohydrate? Period. From all angles. All angles that you can think of, not just analysis. Sam? Some fibers include the microfiber. Very good. So presence of dietary fiber. Some carbohydrates are part of dietary fiber, so they have health implications, such as beta-glucan, for example, is commonly known as a good hydrocolloid dietary, soluble dietary fiber for health reasons. So dietary fiber. What else? Where do we get the most source of energy? Brain food. Yes, energy, brain food. Number one source. Okay, yeah, that's where I was going next. That's good. Number one source of energy. What else? Reducing sugars help with color formation, right? So browning, sometimes browning is good. Sometimes it's not good, but sometimes it's good. Like in baked products, we like to see the browning of the loaf of bread. What else? Anything else coming up? Yeah, that's all right. Yes, through Maillard reaction. What Coleman said, precursor of color and aroma and so on. So functional properties. So functional properties, the starch help with building the body, the texture. So gives you the starch gelatinization adds to the formation of gel matrices, adds viscosity, bulk, holding of the water. So thickening. So if you want the gravy, for example, starch help with thickening your gravy. So functional properties related to structure and feel of the food matrix. Helps with preservation, fruit preserves, jams. High sugar lowers water activity, preserves your products for a longer period of time. and the presence of resistant starch and lowering of the glycemic index. So a lot of benefits for carbohydrates. Of course, there are not so great benefits, like sugars are not good in general. High consumption of sugar, not good for diabetic people. So a lot of energy, not good for dieting, and so on. So there are great advantages. They do have some disadvantages. So what's the importance of analyzing carbohydrates? Let's see. Why is it important to analyze carbohydrates? Do we need them on the label? Do we need what on the label? What's on your nutrition label, Sam? Total carbs, not from analyzing carbohydrates. What total carb by difference, but what's under total carbs? Soluble. Dietary fiber. So you have dietary fiber, that is one of the components of the nutrition label. Sugar, total sugar. Yes. So that is important. So part of the nutrition label panel, some components of the carbohydrate we analyze to get to the nutrition panel. What else? Yes, determine functional properties and shelf life. That's good. We need to analyze how much gelatinization we have, gelatinized starch, retrograded starch, resistant starch. So it will be good to know this. Let me put that quality control. Yes, how much sugar we have that might cause browning, how much reducing sugar versus non-reducing sugar. That's one example. Ingredient authentication. So do we have lactose in our milk, or do we have another source of sugar in our milk? Do we have natural, like honey? Do you have sucrose in honey, or do you have fructose and glucose? Is it synthetic honey? Is it real honey? So ingredient authentication is another importance. And then the compositional analysis specifically for dietary fiber and total sugars, and research and product development. So in research, if we want to determine amylose to amylopectin ratio, for example, because it impacts functionality of the starch, we need to determine how much amylose versus amylopectin I have. Or when we're doing product development, what type of starch we want to use, starch that has more resistant starch in it, or starch that has that starch that can be digestible. So these are just examples of what we analyze carbohydrate for. There's a lot of different methods that we will be covering: analyzing reducing sugars, analyzing small sugars and oligosaccharides, analyzing starch, analyzing dietary fiber. So we will be covering a lot of these different methods. So again, just to remember what different carbohydrates classes we have. Monosaccharides, so sometimes we need to determine what are the different monosaccharides we have. For example, this is important. We did that in our lab when we were looking for a new crop and looking at the different dietary fibers in these crops and trying to classify the different dietary fibers. So we break them down into their singular units, into their monosaccharides, and try to determine what they are, and then figure out what type of dietary fiber we have in our samples. So monosaccharides analysis is important to characterize the different components, as well as determining how much we have of monosaccharides in our sample. Di and oligosaccharides are 2 to 10 units are in this category, and then of course your polysaccharides, starch, dietary fiber, and within starch you have the amylose, amylopectin. Within dietary fiber you have the soluble, insoluble, and within the soluble you have pectin, beta-glucan, arabinoxylans, all these soluble fibers and the dietary fiber insoluble. You have the cellulose, hemicellulose, and lignin. So you have different components there. Okay. So looking at preparing your sample for carbohydrate analysis. I oftentimes in a quiz or exam, I ask you, how do you prepare your sample to analyze for reducing sugars or mono and diglycerides or di and oligosaccharides? And you have a complex food matrix high in moisture, protein, fat. So you want to remove interfering substances so that you can get to your carbohydrates and analyze them. So first step, you want to say, okay, I'm going to dry my sample. And you extract the fat, especially if it's a high fat product. So we remove the fat. You can use Soxit to remove the fat. You can use batch extraction with different solvents. So with Soxit, we use petroleum ether or a combination of petroleum ether and diethyl ether. You can do hexane extraction as well, batch extraction with hexane. But in any case, the fat needs to be removed, water removed, fat removed. That is a general preparation step. But depending on the question that you might get, if I tell you you have like wheat flour or any type of grain, well, we need to homogenize, okay? We need to reduce the particle size. You have to remember that is part of sample preparation. That would be for anything you do, not just for carbohydrate. Okay, so we'll go over total carbohydrate analysis, which is a method that cannot necessarily be used to complex matrices. It's mostly used for beverages that have sugar, small sugar in it, or it can be used for a sample that you have purified. So it has mostly carbohydrate, and you want to know how much in your purified sample you have as carbohydrate. So it's not used as part of approximate analysis. It's not used to determine total carbohydrate for your nutrition label. So this usually we have a lab on that where we get beverages. I think it was 7-Up and beer. Is that right, Rachel? Yeah. Or Sprite? I don't know. Yeah. So what we're going to do for you for this particular lab is Cindy's going to record the lab, like have a video of it, and we'll have a virtual lab session after Thanksgiving where you would watch the video and we'll have data and we'll analyze the data together in the virtual lab session. Because after Thanksgiving, you'll all be home. Okay, so the principle of this method is, we call it phenolic sulfuric acid method because it involves a concentrated sulfuric acid and a phenol compound. The phenol we use is toxic and the sulfuric acid is concentrated. So this is a dangerous lab usually because you're working with unsafe, not very safe reagents. But you're not going to do it this year. Anyway, you're going to observe it. What happens, what's the principle here is, let's say in a beverage, you have sucrose or another source, disaccharide source for making the beverage sweet. So with the sulfuric acid is going to break glycosidic bonds. So when you put your sample in a tube and you add sulfuric acid to it, we usually do that very, very carefully because when you add acid and it splashes, if you add it fast, in a way, not under the hood, it might splash. And that is not safe for you, especially if you're not wearing protective gear. So we do that in a very consistent and fast manner using disposable displacement pipette. What do we call that? Huh? Is that what we use? Positive displacement pipette? Um, I don't know if Cindy is here. I can't even remember what it's called. I don't know if Cindy is here. Anyway, so we use a dispenser, huh? A repipette. Thank you, Cindy. Okay, so it's a repipette. So basically, you'll see it in the video. You dispense about, I don't know, 5 milliliter maybe or more. I can't remember the numbers now, but you dispense a certain amount of of sulfuric acid in a tube, and it has to go straight down in the middle, not on the sides, because once the acid reaches the sample, the reaction starts. It's an exothermic reaction, starts to heat up your sample. So you have heat, you have strong acid, the bonds break. So you end up having monomers. So you will have monosaccharides. Even if you have starch or dietary fiber in there, all the glycosidic bonds will be broken. And you end up with your monomers. The monomers, also in the presence of acid and heat, the heat that is generated from the exothermic reaction, it's going to cause changes in the monosaccharides. We get dehydration, we get enolization. So it goes from your ketones and aldehydes will become furans. So now you have furans. These furans react with our phenol. So you have your where is my here? That's your phenol, that's your furan. They react and you get a substance that absorb at the lambda max is between 480 and 490 nanometers. So it's a spectrophotometric method because we get a compound that absorb light, and what we do is we plot a standard curve. So we have, in the lab, we use glucose as our standard. You prepare glucose at different concentrations. You react the glucose standard the same way you react the sample. You add your concentrated sulfuric acid, then you add your phenol. You have your phenol in the reaction, and what happens is the formation of this compound. Note that this is not a stoichiometric reaction. What that means is the formation of the color or the compound that absorb light, or the different compounds that absorb light in this range, is dependent not only on the concentration, it is dependent on the structure of our furans. So the ketone, for example, is going to give us a different structure than an aldehyde. Then this gives you a difference in compound form that will absorb at this light. Because it's not stoichiometric reaction, the standards we use is very important. So if we know the individual components in our carbohydrates, the best option to do is to prepare a standard curve that has the same ratio or approximately the same ratio of the different monomers in your sample, or potentially different monomers in your sample. So that would be the best way to combat the non-stoichiometric reaction. So moving on to specifically determining mono, di, and oligosaccharides in your sample. So this requires preparation. I'll talk about that preparation in a second. But there are different methods to determine mono, di, and oligosaccharides. We have colorimetric methods, we have chromatographic methods, and we have enzymatic methods. And we're going to talk about the different types of methods. But before that, we need to prepare your sample for such analysis. So if you have a complex food sample and you're only after determining monodi and oligosaccharides, what you will do is you need to get rid of everything else that could interfere in your assay. So in order to do that, first we said we're going to dry the sample. Then we're going to remove the fat by solvent extraction. Then now you have your food matrix that could have starch, dietary fiber, protein, plus the small sugars. So I want to separate my small sugars from protein, starch, and dietary fiber. So one way to do that is by using 80% ethanol to heat, to reflux or extract the sample in hot 80% ethanol. So hot 80% ethanol, your dietary fiber, even the soluble fiber, will precipitate. So dietary fiber will precipitate, starch will precipitate, and protein will precipitate. So what remains soluble in your 80% ethanol are your sugars. So mono, di, and tri, and oligosaccharides. So these will be the only components. Plus, of course, if you have organic acids and small molecular weight colors or other small molecular weight salts and stuff, they will still be with your sugars. So what you do is after if you look at this diagram, so you have your sample here, you put it in a flask, and you have your 80% ethanol, you heat it up, and it would be good to have a reflux system. So you have a distillation unit so that your solvent doesn't evaporate and you don't have efficient extraction. You try to heat it up and have a distillation unit where when the solvent evaporates, it gets cooled and comes down back to your flask. So you enhance the efficiency of the extraction that way. And you will end up, I don't know, maybe an hour or so, you do the extraction and you get your flasks and you have your, you separate your supernatant from the sediment that has the protein, starch, and dietary fiber will be in the sediment. You take the supernatant and you want to remove, what happens is you want to remove the ethanol. So you use the rotary evaporator that you saw in the lab for the isoflavone extraction. Same thing. You put it in a water bath, you heat it under vacuum, you evaporate fast your solvent, and you end up with your residue that is monodioligosaccharides. Then you can dissolve them in whatever solvent you want for the analysis. Could be water, could be buffer, whatever you want, depending on your next analysis. Whatever your method you're going to use, you dissolve them in the compatible solvent. You can clean them further if you want by running them through ion exchange resin so that you can get rid of organic acids, any amino acids that you might have, any charged molecules. You can get rid of that by running them through ion exchange resin, and you end up with a purified sugar solution. Then you can take it further for analysis. Okay, so that's one way. Another way, which is less common, maybe it's more common in Europe because I got this from a European colleague that used to teach here. So it's the Carez treatment. So the Carez treatment is basically a combination of reagents that you put that would break emulsion and precipitate protein, starch, and other dietary fiber and leave your sugars in solution. And it's also helpful, it absorbs colors. So if you have any colors, they get absorbed as well. Okay. So now you have your solution with monodi and oligosaccharides. Then you have to continue with either you're looking for reducing sugar content or you're looking for actual concentration of your different monomers and trimers and oligosaccharides. So there will be different purposes. Let's say you want to look at reducing sugars. You want to determine the amount of reducing sugars that you have. So the common method that we do, even we do it in the lab, is the Smoogie-Nelson method. Smoogie-Nelson is basically the names of two guys that came up with this method, but it's based on reducing cupric ions to cuprous ion. So it is based on the reduction by the reducing sugar of cupric to cuprous in an alkaline solution to enhance the reduction reaction. So the cuprous ion that is produced, uh, this is the equation actually. Well, this is the setup, the old setup where you would have your sample with the reagent and it's blue because of the cupric ion in there giving you that blue color, and this is the reaction. So you have your reducing sugar, you have cupric ion. In the presence of alkaline, you generate cuprous oxide, which is a precipitate and specifically a red precipitate. And it's either measured gravimetrically by weighing that precipitate or by titration. But the method that is most commonly used is the Smoogie-Nelson method, as I said, and that we measure absorption. We have a specific reagent that we use that gives you a specific color. We use spectrophotometer to measure absorption. And so this is what you see, is you see a sediment that is red, but sometimes we see this. This is just an example of something went wrong and you have to go back and think of what you've done, because when you have very dark red and you cannot really distinguish a precipitate in this manner. So what do you think caused this situation? What do you think is different between the upper one and the lower one? Think about when you're preparing your sample. What could you have done that caused this very dark color? You go, oh, I need to change something. It's a simple, it's not a trick question. Pretty simple. Oh, Cindy corrected me. Phenol is added before the acid. Yes, if you don't add the phenol compound before the acid, the reaction won't happen. Anyway, but yeah, back to this. Very simple. Nobody's gonna give me a simple answer. Well, actually, a lot of reduction happened. When reduction happened, you get the cuprous oxide. So a lot of reduction happened. So think the other direction, Coleman. The other direction. You're right on with talking about reduction here. Other organic compounds, other reducing compounds might be present. Yes, Sam? Where you have alkaline pH, it drives the reaction forward. The simple answer, need to dilute. Yes, that's the simple answer. So you just have a very concentrated solution. And this happens all the time in the lab. We start with a concentration. We don't know how the reaction is going to go. You end up having very high absorbances that are inaccurate, or you end up having this situation. So very simple. When you work in the lab, when you get an overreaction that is beyond what you can analyze, you go, okay, I really need to dilute my sample. Simple. Okay, so here's what we are looking at, the different colorimetric methods that are commonly used, and the names that you see are just the names of the folks that came up with these methods, but they have the same principle. So basically, you have cupric ion, you have reducing sugar, and you get reduction from cupric to cuprous. Now, the end reaction is dependent on the different reagents we are using. So what I want you to remember from this slide, not necessarily the different names of the reagents, no, but I want you to know that all these methods are based on the reduction by reducing sugar of cupric ion to cuprous. You need to know this principle. Smoothie Nelson specifically uses I can't even say, arsenomilobdate reagent. So the cupric ion is got reduced to cuprous, and cuprous reduced the arsenomilobdate and gives you a blue color that you measure spectrophotometrically with a spectrophotometer at 520 nanometer. This is the most common method, is to have this reagent and you have your sugar, you have the cupric ion in an alkaline condition, you end up with a blue color that you measure. The other methods, if you can do titration to quantitate, or you can actually gravimetrically weigh your cuprous oxide. So that's what I want you to remember. I don't want you to remember the different names of the reagents and such. So there's a question there that I just realized is not a very smart question. It's asking you what carbohydrates do you analyze using Smoothie-Nelson method and other reduction methods? I just said there for reducing sugars. I don't know why I have this question when the answer is there. Okay, but if I have sucrose, is sucrose reducing? No, it's not reducing sugar. But I want to measure sucrose. I'm determined to measure sucrose using Smoothie Nelson. What do? I break it. So basically, I hydrolyze the glycosidic bond. So I would use HCl acid with a little bit of heat, and I break the glycosidic bond. Now I have glucose and fructose, both are reducing sugars, and I can continue with the reaction and measure the amount of sucrose backwards. Okay, so the next, so these were spectrophotometric, colorimetric methods. The next is enzymatic, but I won't start it because I only have 1 minute, so it doesn't make any sense. So I'll continue. Today's Friday, on Monday. Okay, have a great weekend, you guys.